Hello everybody and welcome to the second video in a series of videos covering exam 2 content. Today we're going to be covering the topic of translation. So to kick this off, what is translation? Translation is essentially just protein synthesis. It's using tRNA and ribosomes to string together amino acids in order to create fully functional proteins. In the human diet, there are 20 essential amino acids. Um, However, there are 64 combination of what are called codons. And these are 3 base pair nucleotide sequences that work together to sort of spell out what amino acids should be coded for. And these are expressed on mRNA fragments. These are degenerate in the sense that there are 64 codons and only the 20 amino acids, meaning some amino acids are coded for multiple times. There's also a starting sequence that's universal in prokaryotes and eukaryotes. It's the UAG methionine sequence, which will begin most proteins in synthesis. And then there's also universal stop codons, which are UAA, UAG, and UGA. Now, um, essentially the way that translation occurs is tRNA will sort of link up or match the mRNA sequence in the ribosomal complex. And while an mRNA may have a codon, tRNA will have the anticodon, which will kind of match it in place. Now, the reason degeneracy kind of occurs is it binds these first 2 segments of the codons very strongly, the first 2 bases. But there's something present called the wobble hypothesis. And essentially what the idea behind that is that the final base pair may have a custom kind of match in the tRNA. For example, like a hypoxyxanthine can bind a G, U, or A in the 3rd position, meaning that while the first 2 bonds are very strong, the last bond kind of wobbles. It's never really completely stable, meaning that it's a little less selective, and that's why some amino acids can have multiple sequences, because the 3rd base pair doesn't totally matter for some of them, just something to keep in mind. So how does translation begin? Translation itself goes in a series of steps. The first step is pre-initiation. Essentially what pre-initiation does is it will combine the amino acid of choice, it can be really anything in this case, with an adenine triphosphate. And that's not adenosine triphosphate, it's actually not ATP. And it will do this through the enzyme amino acyl tRNA synthase. Let me write that one down. Amino acyl tRNA synthase. And this may be one you're asked about, which is why I'm writing it. And it'll essentially combine them together. What this can then do is it can go through a series of processes to sort of merge with the tRNA complex. And that essentially the energy to generate the peptide bonds is actually stored in the phosphate bond with this adenine. So it's interesting. While adenosine can hold energy in these phosphate bonds, so can adenine in this process. So essentially, through a series of essentially 2 pathways, it's just connecting the adenine molecule to the tRNA. You can see it gets shifted over what position it's in, but at the end of the day, they all end up in this final conformation. You won't really be asked on that, but just something to know about your amino acyl tRNA synthases, and that they'll kind of all end up like 2 at the end of the day, amino acyl tRNA synthase 2. And there are multiple checkpoints to check kind of if the tRNA amino acid complex is correct, in which case it doesn't go to translation. So it's kind of like multiple phases of screening. So now let's talk about how translation as a whole, or at least the components of it, are different in prokaryotes and eukaryotes. The first thing is the initial tRNA kind of base that goes into this whole process. In prokaryotes, it's got this really long name, N for methonyl tRNA methionine. Whereas in eukaryotes, it's just methyl tRNA methionine. So much simpler name. They essentially do the same thing, just in the sense that they are slightly differently modified to be a kind of a starting point for the ribosome to work with. And the next thing is the ribosomes themselves. There's a large and a small subunit for both prokaryotes and eukaryotes. In prokaryotes, you have your 30S and your 50S, and in eukaryotes, you have your 40S and your 60S. The way I kind of remember that is eukaryotes are bigger than prokaryotes, so it has the higher numbers. And the last thing is kind of their initiation factors. In prokaryotes, they have IF1, 2, and 3. Eukaryotes have a few more. Like there's an initiation factor 4 as well, but it doesn't go too much higher. So just remember to distinguish those. Also, I'll be using two types of figures throughout this PowerPoint, and I use them kind of interchangeably. I'll try and make a note of whenever I use them. This style of diagram, like right here, this art style is prokaryote, and this one down here is eukaryotes. And again, I'll try and make like a verbal note of that, and I'll try go like sequentially just so that doesn't get too confusing. But I just wanted to point that out now before I really get into it. So essentially, whether you are a prokaryotic ribosome or a eukaryotic ribosome, you have these three little sections that you begin in. You have an E site, a P site, and an A site, EPA. So essentially, this will be where you are working with your entire time. It's kind of like a workspace in a way. In prokaryotes, you initially have sites E and A occupied by initiation factor 3 and 1. This actually blocks the larger ribosome subunit from coming on top, the 50S. Just think of it like a physical barrier, and that's just so if the top of the ribosome goes on before we have any of our tRNAs or amino acids, how's the protein going to get synthesized? That's basically all that is. Something else to note is this what's called a Scheindelgarno region. This is your AUG kind of area, if you remember. That's your start codon. That's the only start codon. So once you encounter this region, your ribosome is kind of set up around that area to where that's essentially in the P site, so it can match the original tRNA. What happens next is that this right here, this N4-methyl tRNA, can bind to this AUG region, and this actually recruits the next initiation factor 2. And all this one does actually is it releases the other initiation factors using a GTP, which is really nice. So now it's just our starting tRNA and amino acid, which is methionine, and that just breaks away. And then once we have that, our large subunit can bind, because if you remember, the initiation factors were basically just preventing that top from going on. We now have our first step. This is essentially pre-initiation and initiation done now. Eukaryotes, it's very similar. We have initiation factors that will bind to prevent the addition of the 60S subunit to the top. In this case, we have our methyl tRNA methionine instead of our N-formamethylneal. That's the essentially only difference there. It's very similar in the fact that there's there's EIF2 and EIF3 instead of just IFE. This is kind of just a denotation to like be eukaryotic. So like whenever you see that, it just means this is in eukaryotes. Same kind of thing. Very similar process here. The only thing is there's actually in eukaryotes also an EIF4, which will essentially detach the complex as well. There's still an EIF2 that'll do the same. So a little bit complicated, but all it's doing is doing the same thing. The factors are initially bind to prevent that top subunit from binding. We have our initial tRNA which binds, and then through EIF2 and EIF4, the complex is released, and then the top subunit can go on. And it still uses a GTP for energy as well. So very similar. It's mostly just nomenclature that differs between these two, as long as one other eukaryotic initiation factor. So a really quick little side note, Dr. Phoenix likes to have kind of little therapeutics questions thrown in about certain drugs. One of them he wants you to know about is streptomycin. I don't know if Dr. Zhang will do the same thing, but in case he includes it, I'll just mention it here. Streptomycin prevents the essentially the binding sites of the 30th subunit. So the ribosomal complex with 30th and 50th can never be assembled. Which just prevents initiation from starting and will prevent protein synthesis. So that's how that drug works. You might see a question on it. I think it was on a quiz for us, maybe not the exam, but it's just a good thing to know. Okay, so now let's go into the next step of translation, which is elongation. I'll be starting with a eukaryotic diagram, and then halfway through I'll be switching to prokaryotic, so I apologize in advance for that. I'll let you know when I do. So what essentially happens next is we have our amino acyl tRNA methionine. It's happy in the P site. What we can do now is we're actually going to add an amino acyl tRNA to the A site. Our methionine one will still be in the P site for now. We're just going to use GTP in EEF1A or eukaryotic elongation factor 1A and a GTP molecule and use some energy to put this into our A site. So now it's right next to our P site. They're both in. There's no protein chain that's been synthesized yet. They're kind of just resting right next to each other. But we can use this enzyme right here, peptidyltransferase, which will actually move our methionine, which was up here, and actually stack it on top of valine and make a peptide bond. And if you remember, the phosphate groups are what gives the energy to the peptide bond as it's forming. This is very similar in prokaryotes. Instead of elongation factor 1A, they have elongation factor 2, spelled TU. Something else to note also is that these tRNA systems, or these elongation factor systems rather, are reusable. Even though they use up a GDP or a GTP and turn it into GDP, by cycling around the cytoplasm and exchanging amino acids, they can actually cycle through and be reused just so we don't constantly have to make those. So that's really handy. So here's the part where I'm going to be switching into prokaryotes. After we've used our peptidyltransferase, you can see now we have something in the P site and the A site. So ideally what we'd like to do is as you might be able to see, we want to be able to like make this process as repeatable as possible, but we're kind of at a stopping point in the sense that we move something from our P site to an A site, but now we're all in the A site. So the logical thing to do would be essentially to see if we can get our P sites and our A sites to shift over one, and then we can keep the stacking process. And that's exactly what happens. So essentially, we have in prokaryotes, in this diagram, a factor called EFG, elongation factor G. And this will use GTP, and it'll bind to the A site, actually. And what this does is it'll move our P site and our A site over to our E site and our P site, respectively. So now our protein stacks in our P site. And then once this GTP has been used up by EFG, it will essentially release off into the cytoplasm. And along with that, our empty amino acyl tRNA, which was originally the one with methionine, will also just dissociate. So now we're kind of back to square one, just with instead of solely methionine in our P site, now we have a methionine valine chain. And then this will repeat until termination occurs. And also, instead of EFG in eukaryotes for this step, the factor is elongation factor 2, or eukaryotic elongation factor 2. There we go. And this will repeat until it needs to stop eventually, and that'll be the next step. That'll be termination. Something else, though, that I do want to note, in prokaryotes especially, This can happen multiple times at once since they can essentially, if you may remember from our last lecture, their mRNA may be like an entire promoter region of an operon, and it'll form these complexes called polysomes just so we could make multiple protein chains at once, um, which can be really useful on a simpler scale for proteins that are kind of right next to each other, just to constantly be producing things that are needed for the cell. Okay, so as I alluded to on the previous slide, the final step in translation is termination. This is probably the most basic of the steps. Something called a release factor, and this happens in both eukaryotes and prokaryotes, binds to the A site when the A site is empty. So as we established on the previous slides, when the A site can be released. And essentially what that does is that releases our protein as well as all the components afterwards that were used in translation. So we have our ribosomes, we have our mRNA transcript, and we have all the tRNAs and factors, which are all reusable since they're all proteins to be where they're needed. But most importantly, we have our protein chain. But this isn't necessarily the end of the road for the creation of our protein, because certain groups can be added to it, it can be folded in certain ways, parts can be cleaved off, kind of just depending on what we need it to do. And there's 3 sites where this happens. We have our nucleus, we have our rough endoplasmic reticulum, and we have our Golgi apparatus. And I would say for this class, really the only one you need to kind of know a little bit about is the rough endoplasmic reticulum. Essentially, as this protein is being, or this protein is being produced by like a ribosome, it may pass through the rough endoplasmic reticulum. In fact, it most likely will, because if you remember, while there are ribosomes in the cytoplasm, most of the ribosomes in your cells are in the walls of the rough endoplasmic reticulum. It may essentially find a receptor that essentially brings certain parts of the protein into the cell and can cleave them in various ways. Like in this case, this part gets cut off and this part goes into the cell. This is folded. And this is just all in the membrane of the rough endoplasmic reticulum. So these proteins can be further modified, especially if they're made at this site, to be just folded, and then they can be exported to other parts of the cell. So really quick, we have a few more drug examples, except these are more like elongation drug examples, whereas streptomycin was an initiation drug example. In the case of puromycin, this will block the A site of ribosomes, which means no amino acyl tRNAs can go in and form the protein chain, meaning essentially in prokaryotes, which we use puromycin in to combat, they can't make proteins. Next, we have our macrolide antibiotics. A few examples of those are azithromycin, clarithromycin, and azithromycin. Not streptomycin, that's actually an aminoglycoside antibiotic, which is why it's on a different slide. These will actually block the 50S subunit and kind of prevent things from moving. The step where we essentially move everything to the from the P site and A site to the E site and P site. And then very similarly, we have the strept of tetracyclines, doxycycline, binoxycycline, and these other ones. These will block the 30S subunit, kind of like streptomycin did, but they're a different class, and they're more blocking how kind of the 30S subunit interacts with the mRNA. Tunicamycin is a different class. How this one will work actually is it'll block the post-translational folding in the rough endoplasmic reticulum, kind of like we talked about on the previous slide. It'll just prevent the proper folding, and it can mess up some of the final products of proteins. So not really stopping at the beginning, but more in the post-translational modification step. And then finally, this isn't so much a drug, it's diphtheria toxin. It'll block the binding of eukaryotic elongation factor 2. So this isn't like an antibiotic because it's in eukaryotes. We're obviously not really trying to harm our own bodies by this. And this essentially works kind of similarly to how the macrolides work in us, is it blocks the displacement of the P sites and A sites to the E sites and P sites. just in eukaryotes. So that's obviously not something we want in our body, because that's very similar to what a macrolide does to a prokaryote, which kills it. We don't want to kill our own cells. And with that, I think that's everything on translation. Thank you so much, and I'll see you in the next one.